to introduce Martin Tournier, who you guys pretty much all know, at least all of you in the room.
Yeah, and it's mainly the first time here.
So he gave a talk here about a year ago where he was talking about his PhD work. So today I will give another short intro, very similar to what I did before, maybe a little shorter because I forgot I needed to do it until 5 minutes ago. But Martin got his first master's in oceanography and marine environment, and then he realized he needed more statistics, so if you guys have questions, he's a good resource. And he went back and got another master's in modeling and ecology before he went and did his PhD at La Rochelle University, where he was doing habitat modeling of deep diving marine mammals. And that is some of the work that he presented on about a year ago, some of the work he did with a sonar tag with elephant seals. He joined our lab last January and has been part of the Emperor Penguin Project along with helping with other things. For example, he helped teach the first biologging course that we taught last fall, and then he was part of the team that went to Cape Crozier in November of last year. And I think he'll show some videos, so even though that's not the data that he's analyzing, he helped with that data collection. I think he'll share some of that. So with that, I will pass it off to him, and he can give you a little update about what he's been working on.
Thank you, Gita. Thank you for this nice introduction. So, hi everybody, hi Vertlab, and hi other people. So I won't insist on presenting myself for sure, but today I will be presenting some of my work, focusing on the Ross Sea emperor penguin foraging behavior. So the first part will be really a presentation of my work, and there is a second part, a more casual, that is a little summary of our fieldwork campaign last fall in Cape Crozier. To start with the emperor penguin, so who are they? So you can find them all around the Antarctic continent. Their total population is estimated around 200,000 individuals. Here you can see this water body is called the Ross Sea, and I will talk a lot about it today. And it's where you can find around 25% of the total emperor penguin population. And we, to date, identified 66 colonies of this species, thanks to some remote sensing approaches. So the emperor penguin is an iconic species of the Antarctic continent, and it's also iconic because it's mainly threatened by global changes, and latest studies suggest a decline of the population, unfortunately. So they found that over the last decade, the global population was decreasing by 1.3%, so around 10% over the decade. But we don't know the variation at the colony level. Some areas are showing some increased trends, and some other areas mostly a decline. So to have better model about their demographic dynamic, we need to integrate those small-scale variation. And their decline is mainly due to major reproduction failures rather than a constant decline. And today we'll talk about the penguin in the Ross Sea. So why those populations are important? Because the Ross Sea will be their last refuge. As you may know, we're facing climate warming, and so we can model and project their demography over the time, and with the most optimistic models, by the end of the century, the Ross Sea will be their last refuge, or one of the last refuge of them. And under the most pessimistic, it might be also their last refuge, but yeah, it's our hard to tell more with those models that are only integrating the global trends and mostly the sea ice variation. So now I will introduce you the emperor penguin life cycle. So I will break down each step over their life cycle. So what you may notice is in blue on the circle is all the phases that are happening at sea, and in white all the phases that are happening on the ice. So just notice that their breeding cycle is reversed from what we know on common bird species we are. So they're mating during the fall, and they're incubating over the winter. So just be aware that we're talking about the southern hemisphere, so the summer is around January and the winter around July, August. So from their mating, so we'll start by the mating period. So around early April, so there will be a courtship and then a mating. They need for this a stable, fast ice. I will come back on the ice vocabulary just later. So I will define all the terms here. So they need this fast ice from April to December. And for this, they're coming back from the sea, and they need to walk from the ice edge a few kilometers to 100 kilometers, depending on the colony they're living. So yeah, the life cycle timing can show some local differences between the colonies. So after the mating, so the incubation is around 60 days, and then the female will lay an egg, only egg, and so around mid-May, and they transfer it directly to the male. The females are going back to the sea to forage and replenish their body condition. And the male will incubate the egg. And so the total period from the courtship to the hatch of the egg, it's over 100 days. And so the male will fast over this period. And while it's in winter, so when they are hit by some snowstorm or very rough cold, they will huddle to conserve their warmth. Then you have the hatch and then the early chick rearing. So from this period, there will be always one parent taking care of the chick. They will stay on one parent's feet, and the other parents will be at sea forage, and they will come back to feed their chick. And that is happening from September to November, and at the end of the season, both parents can be at sea foraging. The chick is left alone, and if they're hit by a snowstorm, they can huddle too to conserve their warmth. So this is a well-known feeding here of a chick. So it's a perfect example of what we call the trophallaxis. So the adult is regurgitating the food to feed the chick. And then in December, you will have the ice breakup. And so from this point, the parents will stop returning, so they will stay at sea foraging for themselves. And then the chicks are moving together to the ice edge, and they're fledging, so departing at sea together. And for the adults, in January, they will, so for the Ross Sea penguins, they will mostly travel to the eastern Ross Sea, where they will go back on ice, and they will fast for 1 month, and they will be performing their catastrophic molt. So they're losing all their old feathers and regrowing new ones. So during this period, they cannot swim, and they will lose almost half of their weight during this period. So as you can see, the penguin life cycle is highly dependent on the ice dynamic. So here are some definition of those ice features. So the fast ice, or the land fast ice, is the sea ice which is fastened to a continental land body. The pack ice that are large pieces of floating ice together into a nearly continuous mass. Ice floes are some segment of pack ice that are floating and that are larger than 20 meters. And polynyas are area of open water surrounded by sea ice. And during the breeding season, the emperor penguins need fast ice to raise their chick. And during the molting season, they need either pack ice or fast ice to be able to molt. And the sea ice dynamic determines also their breeding success. So there is a direct relationship that was shown in some studies between the distance to the sea ice edge and the colony location. So the further away is the sea ice edge, the lower can be the breeding success. They also strongly rely on small spatiotemporal scale feeders. So they will use polynyas or sea ice crack to be able to jump at sea before reaching the sea ice edge. Sorry, I did not define the sea ice edge, but that will be here. So it's defined as the location where the ice concentration, the sea ice concentration, is lower than 25 to 15% of the whole sea coverage. So, sorry. And so, coming back to the ties between the sea ice dynamic and the emperor penguin breeding success, the sea ice dynamics simply drive the marine food chain. So when your sea ice is melting, it releases some nutrients, and the nutrients are enhancing some primary production and then supplying the food chain and so on. For our Cape Crozier colony, the sea edge is close by. So for them, the foraging success might be less driven by sea ice dynamics, but mostly by the marine resources that are around. And just to show you, there was a recent major ice breakup, mostly around the Antarctica Peninsula. So for some Antarctica Peninsula colonies, there was a total breeding failure because the fast ice break up during the chick rearing season, and then all the chicks drown. And so that was 2022 and a zero recruitment year for those colonies. So the sea ice dynamic is important. Why? Also part because they're performing a foraging at sea. So this is their tracks. So they're foraging trips during the post-molt season. So by the end of the summer to the mid-fall. As you can see, they can cover the whole Ross Sea. So the breeders will come back to some colonies around here, and the non-breeders will go away, sometimes really far from the Ross Sea. It's a very different situation during the breeding season because the parents are feeding their chicks, so they're depart the colony, they go at sea, forage here, and come back. So you can see the geographic scale is really lower during the breeding season. So that implies that they are using different foraging ground and you're mostly using different foraging strategies during those two at-sea trips. And it's important to take into account the third dimension of the ocean because emperor penguins are deep diver. So here are the record, the maximum record we have. They can reach depths of 564 meters deep and a duration up to 32 minutes. Was not the same dive, but just to give you the extrema. But you will see mostly dive around 70 to 90 meters. So this is a mean dive depth, 70 to 90 meter deep, and the mean duration will be 4.5 minutes. And so why they're diving? Because they're finding their food there, mainly compounded by the Antarctic silverfish, but also it encompasses krill species, crustacean, some cephalopods, so a diverse diet. And so as you can see, all along their foraging trip, they're diving at different depths. Here it's a trip from Cape Crozier around the Ross Bank, which is this feature, and coming back to the colony. So it's important to take into consideration that their foraging environment is in the three dimension of the ocean. So their breeding period foraging trips, as I told you, they're small scale, so their duration is in between 4 to 10 days. For a distance to from 300 to 1,000 kilometers. And their foraging habitat and the habitat use of the breeding emperor penguin in Cape Crozier is the focus of the work of Caitlin Kroger. And for my part, I'm focused on the post-molt foraging trips. So here they're very different. Their duration is 2 months for the successful breeders of the next season, and up to 6 months for the non-breeders. And the distance can be up to 9,000 kilometers for the non-breeders. And so that characteristic of the foraging trip after the molt led me to, so I'm working on two driving questions. The first is, what are the environmental conditions that are maximizing their foraging behavior and foraging activity? And the second one is to know if they are displaying different foraging strategies in between the individuals. To answer those questions, there is a powerful tool that exists. They're called habitat use model. So you may know the species distribution model. So in ecology, they're widely used. It's the exact same statistical background. The only difference is the research question, which is slightly different. So to build those models, we need two data sources: the presence absence of the species, or here the tracks of the species, and some environmental data sources. So let's start with the tracks. How do we collect those? So there are two different approaching. Either it's the post-molt or the breeding season. For the post-molt season, how to approach the penguins. So they will be on pack ice, so ice floes or fast ice. And so you can approach them only with a boat because it's far remotely on the eastern Ross Sea. So it's the easiest way to be close to them. And for the breeding season, it's different because it's during their chick rearing period. So the penguins are on fast ice in their colony, and we can approach them on foot. and then 2 different loggers are deployed in those 2 different periods. So the splash tag for the post-molt individual, so it's a deployment only. There will be no tag retrieval. So a rule of thumb to know if a tag is, it's not always working, but if the tag will be recovered or not. If you have an antenna, it will, like a large antenna, it will say that it will transmit data to a satellite. So it's a satellite relay data logger, and it will be no recovery of those. Can be, but they can transmit data. For this one, you don't see any antenna because we need to retrieve them to be able to collect the data. So, so they're also collecting other information, like the depth, the diving depth the penguin reached, or the duration of their dives. And for the AxiTrack, we have more data collected. And so the main difference between those data, the post-molt location data will be Argos position that are coarser, so they are less correct, I'll say, to the GPS position that are more accurate. And then the data are satellite transmitted, so I can have only a summary of their foraging activities, so a summary of their dives, so mostly the deepest point of their dive and the duration of the dive. For the AxiTrack data, we can collect a lot of different information, so they can collect up to 1 point every second of their depth, time, and also their acceleration in the 3D environment. So that is the main difference in between those track data. So once you have your track data, how to build your model. First, you will need to clean or filter those tracks because you will have some outliers. How to filter them? So you can use a speed filter or a net displacement filter. So net displacement is just saying, hey, this point is close to Australia, it's a bit far, so I will remove it. The speed is the same logic. To reach from this point to this point, it took only 2 minutes, is way too fast. We discard this point. So once you've done this filter step, you will need to do a post-process because the algorithms like really regular time grids, and it's easier to handle the data like this over a consistent timestamp. We'll do this post-processing step using different algorithm. I was using a state space model, and so it can also segment the track, and with the information bring by the track, it can say what was the behavioral state of the animal during this track segment. It can be either a rest period, it can be a foraging segment, or just a traveling segment. So you can be able to type your travel segment doing that. So the second step will be to simulate the tracks. So because in those models you need presence data, absence data, you need to know where they did not go, but where they would be where they will be able to go, but where they did not actually go. And so to do that, you extract the characteristic of your track, the turning angle, the speed, segment distance, and so on. And then using those characteristics, you can simulate other tracks, starting from the departure point and ending at the same ending point. And then you can collect a random number of data points that will be the absence point of your model. And then you need to collocate environmental conditions for the observed and simulated points. So here, for example, I will collect for all the covariate I will select the value of the environmental covariate under the point, so the location point of the animal. Here is the sea ice concentration, for example. So talking about the environmental variable, which is the second data source you need, it's pretty challenging here because you're in the southernmost water body of the world, and so it's really hard to obtain environmental data under the 77 south parallel. And the main problem leading that is that you don't have a big data collection there because the polar orbit satellites are sampling a really narrow bands over the Ross Sea, and there is often a big cloud coverage, or during the winter you will have a large sea ice coverage that prevents the satellite to collect the data. For the robotic samplers like profiling floats, they will be mostly trapped under the ice, or they will be trapped in the no current zone. And for the shipboard measurement, you will have, it will need heavy logistic. So that's why we don't have a lot of data collection in this area. So the only solution to fill this gap is to use model outcomes that assimilate many different data sources from everything I told you just now, and they will combine them and try to interpolate the value of the environmental covariate. So I started first to look at the regional ocean modeling system, the ROMS. It's a pretty nice model that gives a really fine resolution data down to 1.5 kilometers. The problem is this model was not run for this region after 2014, so it's not possible to use this one. Then I look at the global reanalysis model that you can find on the Copernicus repository. So it's a global model, so it's made for all the oceans. You still have a hard line at the 77, so south of the 77 parallel, and a limited number of variable. But the good thing is they can have some data profile, so you have at depth value. And the best I could find is the Southern Ocean State Estimate, the big SOSI. So it has a coverage down to the 78 south parallel. It's designed locally, so it's a model specific to the Southern Ocean. It has a lot of different biochemical outputs, which is great, and at depth value too. But the only drawback is that it has a coarser spatial resolution. So I need to be still aware that it can be extrapolated under the 77 parallel because the model is based on a very few data points, but it's better than no data at all. And then what are the covariates I can integrate, and I am integrating now in the habitat use model. So in those statistical framework, you have two main data types. So the static data, so like the bathymetry. They're static because they don't change over time. It's pretty nice, but you don't have a lot of those. So for the bathymetry, most of the model over Antarctica for species living around Antarctica, they're taking the depth, the slope value at the bottom, or the distance from the shelf break. And then there are a lot of dynamic covariates we can use, so mostly the sea ice, because emperor penguins really rely on sea ice. It's important to integrate different kind of sea ice covariate. I compute, for example, the distance from the sea ice edge for any location, or a metric about the sea ice variation over time. That can be also insightful. Then the water temperature, like the sea surface temperature, the temperature at 100 meter deep, the depth of the maximum temperature of our profile, and so on, and also some biological covariate, like the chlorophyll A concentration, or also there is a nice model on the necton abundances that goes down to the Ross Sea. And so all those covariates will be and are integrated in this habitat use model. And some more too that need to be tested. So the modeling approach, so as I told you, it's always the same. So here it's an habitat use model. So rather than a presence-absence point, it's the response variable is the presence or the number of dive per location or per hour, and it will be fit over the values of each tested covariate. And then to fit your model, you have the choice in between different model algorithm. Most of the paper you will read about habitat model are using GAMS or boosted regression trees. So I am testing both of them too. And then what are you obtaining at the end? You're obtaining maps like this, and the colors represent the probability. So here it will be the probability of a foraging event given the set of environmental covariate values at X, Y, Z, and T, so at a proper location in a specific time. So what are the preliminary results? So I don't have a lot of results yet, but those are the partial dependence plot that I have right now. And so how to read those partial dependent plots, because you always have those when you're using GAMS anyway. So here is your covariate, and here on the y-axis is a scaled proportional increase or decrease of your response. And so it means that if you're here, your foraging activity will be high here, and here will be low. So as you can see, the model states that under a bathymetry of 500 meter deep, your foraging activity is higher. It's maximized for a temperature around -1 degree Celsius, and for chlorophyll A concentration around 1 milligram per cubic meter. And so the first model runs show the importance of sea ice variables. So I can show you here right now just the rank of the importance, but still the sea ice variables are the most important variables that are driving their foraging activity, but also some biological components are important. But sorry, I don't have the model maps now to show you. but it will come soon, and I will be happy to share it with you soon. What we can look at is another study back in 2015. So it was a habitat suitability model for the emperor penguin. This model was excluding the Ross Sea, as you can see here. And they did this habitat suitability model for the breeding season and for the pre-molt season. And here, the more red, the more suitable it is for the emperor penguin to forage or to be. And as you can see, for the breeding season and for the pre-molt season, the habitat suitability is very different. The main driver of the breeding season is the distance from the colony, because the emperor penguin can go as far as they want to forage, so they need to stay close to the colony to be able to come back fast to feed their chick. It's not the same constraint during the pre-molt period, and here it was mostly sea ice covariates that were driving that was driving the model output, and also the presence of the polar front. So as you can see, it's pretty the similar shape of the polar front. So that can be different habitats because they're not on the same location, and applying different foraging strategies in between those periods for the emperor penguin. So we are hoping to do the same, but with different variables for a different location and with more environmental layers to test too. So, sorry, that was the end of the proper work. And now it's time to give you a little fun summary of our Cape Crozier 2024 fieldwork. So here is the picture from the ILO, and you can see, so this is the eastern tip of the Ross Sea. Here is some blue fast ice, and in this first little crack was our campsite, and in the third crack here, it's a bit stained by poop, by bird guano. That was the colony that was there. And then, so how was the work there? So that was the penguin highway. So here was the colony, and then that was the way to the ice edge. So, and we were capturing the birds this way, so they were at their foraging trip, and then tagging them in a short procedure. It was under 30 minutes, so it was quite quick. And when they were done with their foraging and they were coming back to the colony, we tried to capture them here. So here is the proud little guy with the two tags. So this is the AxiTrack tag that records a lot of different information I just showed you before. And this is the VHF antenna, and it was really important to find them. So we were scanning with the antenna every 15 minutes, and if there was a beep, we were running to try to catch him before he reached the colony, because then if he was in the middle of the colony, it was a pretty nice game of where is Waldo, but with a penguin, and so if the penguin was facing you, you could wait hours before finding him. So yeah, it was easier to catch him on his way back rather than in the colony. And then we also enjoy the downtime to collect guano sample, and it was done under very strict surveillance of the penguin, so it was really done properly. And so those guano samples will be used for a DNA analysis to understand their diet. But we also enjoy and witness some really cute moments and behavior. So that was really, really cool. And some preliminary result, or just to show you the track of those penguins. As you can see, so over 20 tags deployed, we recovered 15. So it's a good 70% success. That's really good. And so as you can see, most of them, or all of them, went to this Rose Bank feature. So a shallower shallower feeder where they're all foraging. But there was one guy that just did a different thing. He went really close to the colony, which is here. Here actually it's 2 trips, 2 different at-sea trips, so over 7 or 8 days. So he stayed 3 days at sea. He was able to gain a lot of weight, forage a lot of food, was able to feed each chick and go back to the sea. And so he displayed a very different behavior than all the other penguins. That was pretty cool. And then the life at the camp. So this is the campsite with the different tents from a different angle. So the sleeping tents here, pretty cozy and warm. And the blue tent was a lab tent, cooking tent. So yeah, many, many different uses. Was really cozy and warm, except at the end of the season, we were hit by a violent storm, a snowstorm. But hopefully we learned how to make snow walls, so we were safe. But the only mistake is the snow wall shouldn't be so close to the tent because it will cover you and bury you under a big amount of snow. But hopefully everybody is safe now. So that was, but most of the time, we had a really lovely weather, and it was really sunny and actually quite warm. That was great. And to bring you some experience of emperor penguin colony, we're using this 360 camera inside the colony to record some behaviors of the penguins. And here is a small video with different displays of different behavior, chicks calling for their parents or parents calling for their chicks, some human-induced behavior, you will see that, and yeah, some cuteness. Thank you all for your attention. Yes, Amber.
I know that that main map was Parker's, but it kind of looked like the penguins were doing a lot of dives to the bottom. Is that actually true, or is it just because the bottom is kind of, the bathymetry there is kind of?
Yeah, so we can go back to this slide. Oh, no, you don't see.
I was just asking if the penguins do actually dive to the bottom, or if the bathymetry is just tricking me from this photo.
So you're right. There are some dives that are piriform and that can reach almost the bottom. So they did, how to say? They used the metric, and they were saying that dive was a bottom dive, so or even a benthic dive, if the maximum depth of the dive was at least 80% of the depth of the bottom, of the bathymetry. So yeah, they're performing actually bottom dives, or almost benthic dives, and also some more pelagic dives. Yeah, they're displaying the two.
Do you know if anything they're eating is actually buried in the sediment? Because it seems like, from what I thought, they're mostly eating like pelagic fish.
I know those fish are found pretty deep, actually. At least the adults are found pretty deep. They're the main resource of many predators in Antarctica. And they will have some demersal life, so not at the bottom, but close to the bottom. They're not like not in the sediment. I don't know about, because there are some target species that are crustacean.
Oh yeah.
I don't remember if there was some target species that are bent.
So the euphausids are probably in the water column. I think, and the silverfish are also. Hopefully, Vicky will have a defense on this in a little bit, because right now we think they mainly eat euphausids and silverfish, which are found in the water column, but we do know they dive to the bottom, and so hopefully Vicky will be able to answer a little bit about what they're, because not our research group, but a group on the cruise was doing tows on the Ross Bank to actually, they were looking for corals, but anytime they accidentally caught a fish, we kept it. So there are other species down there.
I know those fish are kind of nesting on the ground, because they can I remember those studies, they were scanning the bottom of not this region but around Antarctica, and they were seeing pretty round shaped feeders, and that was corresponding to those fish called nests, and they were counting their abundance like this. So they are living close to the bottom of the sea, at least in one period of their life cycle. But yeah, I don't think they're bury themselves in the sediment. But yeah, I think they can find them close to their nest and try to eat them there.
Great job, Martin. Great presentation. I actually understood the stats and model making that you were doing, which is not something I can always say for presentations like this about tracks and stuff like that, and data logger data. Sorry, can you go to the last couple graphs with your GAMs that you had?
Sure.
I had a question about one of them. I thought it was interesting that it seemed like they were foraging more in areas where the chlorophyll content wasn't at its the highest found, and actually they were foraging less in areas where they found less. And then, sorry, two-part question, that, and then also how does this data that you've found so far kind of relate to that paper you discussed in 2015?
Yeah. So it's a good question. I think they have a pretty good name because they're partial dependence plot, and so they're just capturing partially what happened. Maybe, and most probably, in our data set, we didn't have a lot of data points that were really rich in chlorophyll A. So those values of chlorophyll A are really underrepresented in comparison to those values, like mid value. And so most probably the foraging behavior was higher here because most of the location show values in between those two. So it's a partial capture of what is maximizing their foraging, or actually maximizing display of a foraging behavior. But usually we show some lines here that show how dense it is in terms of values of the covariate.
I'll pass it off to someone else, but I have something that builds on that. Could you include like a time factor? Because the birds aren't eating chlorophyll, they're eating zooplankton. So could you maybe do something like a time lag, like time after chlorophyll?
Yeah, I think it's what I want to do now, and I was doing that before for other models, is the chlorophyll A, it's quite important in terms of dynamic because it will lead the start of the food chain, but as you said, they are not feeding on chlorophyll A, and there is some integrating time in the food chain. So I think it's around 2 to 3 weeks before seeing like a big bloom in necton. So from zooplankton, so from primary predator to zooplankton that feeding on, are grazing on on phytoplankton, and then from the necton that is feeding on the zooplankton, and then this necton is the diet of the emperor penguin. So I think it's very interesting to add dynamic covariate of the chlorophyll A.
Hi, thank you very much. This was really awesome. I loved the pictures at the end as well. I appreciate that in seminars. I was wondering, at the beginning you said that the colonies around the Ross Sea would be the most resilient into the latter half of this century. I was wondering what ecological and or physical conditions are making these Ross Sea colonies so resilient, and why are they predicted to last the longest?
Good point. First, this is the southernmost water basin, and here the sea ice dynamic is pretty stable over the years compared to, for example, the Antarctic Peninsula. So that is the main reason that it's considered as the refuge. I think it's mostly latitude and where the ice will be the more stable because it's far from the main currents that are circumpolar, and so the water circulation is lower, so the heat circulation is also lower. So I think it's really the main reason why it will stay a refuge by the end of the century.
Awesome.
That was maybe some other factors, but I'm not aware of.
Great, thanks.
All right, nice talk, and also very cool field work to be a part of, right?
Yes, no, really.
So the B-SASI model is one of the key parts underlying the habitat model you're creating, and as you mentioned, there's not that many observations next to the Ross Ice Shelf. So I'm curious, are there any measurements that are in the tags that you deploy that could be used to compare to the model that might tell you like how well the model is doing in that area?
So not for those tags. I mean,
It was, but it didn't work.
Yeah. So, but yeah, those talks, because they're giving you a summary of the dive, so they're actually recording the temperature. I don't know how trustful they are, but it gives me only a summary over the dive, so I don't have any profile, temperature profile, so I can use those.
We asked the tags to send a profile. 'Cause
Oh, yeah.
They did actually have an instantaneous
Yeah.
temperature profile, and they are very accurate, like we've calculated, they're very good, but for some reason, they did not transmit
Yeah.
any data.
Oh, yeah.
Even though we requested that they would.
Oh, sorry, yeah.
No, no,
I
it's fine. You
forgot
weren't-
that. Yeah, yeah.
I was the one that programmed them, so
Yeah,
you're seeing that the data doesn't exist.
Yeah,
but
yeah.
We did ask it to send, like it would take 8 data points on the, like every 6 hours, it would take the deepest dive and it would transmit 8 data points that could profile it. It just didn't work. So.
And
The last I checked for BSaucy, it was run till like 2023, I think. Did they run it further into 2024 to overlap with the field season?
no, but what I did not tell you is that I'm taking only a part of their foraging trip to correspond to what the breeders will experience, and also before the polar night. So the polar night is my threshold here, and I'm analyzing everything before, so I don't need the data from 2024. And there is only, I think, yeah, there is no penguin that reached 2024 because the tags were deployed in February. So yeah, it will be a long record. They all lost the tags before that. Almost December, I think one like reached December, but no 2024.
Thank you. Thanks, Martin. That's all really fascinating. I learned a lot. I have so many questions. So I don't know if these fit within the scope. This might just be like general information question. But when the penguins go out foraging, you talked about a range that they can, swim out and go forage. Is that physiologically limited? And I'm thinking specifically, I thought of it when you showed those 2 plots side by side of the breeding season compared to the post-molt, I think it was. And I mean, there was that band that was heavily used that was just offshore. Yeah, the last one where it was sort of an integrated, a little later. But that made me wonder, is that where the sea ice was? Another. Yeah, it was from the other paper. Sorry. Thank you guys.
Sorry.
Yeah. So I mean, that band in the pre-molt, is that related to where the ice was at that time and that's sort of the distance that they might travel, or is that behaviorally where they want to go? I'm trying to wonder if there's like a limitation of how far they can swim.
Yeah, so actually, so this habitat suitability model is built on many different tracks for different colonies, mostly from the other side of Antarctica. And so there was also where there were the denser foraging activity. It's pretty overlapping pretty well with the polar front, where the polar front is, and the polar front is known to be a physical barrier that is concentrating a lot of different prey them. So that's why it will be here where you will find the highest foraging activities. And I don't remember why this buffer zone, kind of buffer zone, I don't remember the drivers of this buffer zone. Yeah, sorry.
That's okay, but it almost seemed like with yours where they were swimming out to that deeper area that they also were cruising farther away from land than they needed to, other than the one penguin that stayed near shore. So then my following question is, like, energetically, is there a cruising low energy swim mode for the penguins to go out further offshore to those better foraging sites? Is it?
I think the I'm not really good on the energetic questions about that. But I think it's really the trade-off between how fast I can forage and accumulate a lot of food to feed my chick against the energy consumption I need to put into the swimming to these further areas. Unfortunately, oh sorry, I have just a partial view on this because we don't have the depth data or the depth data a weird thing, and we can use them right now. So it would be interesting to see how deep this guy was going and how was the frequency of his dives. But yeah, I'm not sure about the trade-off that is behind those two different strategies. Yeah, I think maybe Gita has more answer about that. No.
Yeah, well, we did not think they were going to go this far.
Yeah.
So we knew at Cape Washington they went out to the Prairie Bank, which is closer, like 150 kilometers, like a lot of them did. And so we actually hypothesized that birds here would be doing something different because that the Ross Bank would be too far and it would be beneficial. If they heard this rock, they all went to the Ross Bank.
Right.
So obviously the food there is beneficial enough to make it worth going. And they probably are eating like on their way out and on their way back. And the way back is probably where they're leaving the chicks. But like in other areas of Antarctica, they often feed on the shelf. They don't actually dive as deep as they do in the Ross Bank, but the Ross shelf is pretty deep. So, but yeah, our hypothesis was that birds here would behave differently than Cape Washington, and most of them would just forage in the water column above deep water. But
Did not. Yeah.
And I think also it's a matter of predictability. So because only the fact that this guy went twice to the same area, I think there was like a high predictability that if his first trip was successful enough, it would be good to just stick with this strategy. And maybe it's part of the answer here. You want to target a very predictable environment where you are sure to find enough food. And so like this, you know, I mean, yeah, I don't want to overthink this thing, but yeah, I think predictability is could be one part of the answer here.
Yeah. Neat. Thank you.
there's something else. Thank you very much.
Thank you.